the Pennsylvania State University in 1974 and obtained his PhD in biophysics under supervision Dr. Strode at the California Institute of Technology in Pasadena. Later, he worked with Dr. Martin Raff from 1981 to 82 at the University College of London. Dr. Martin Raff is one of the authors of Molecular Biology of the Cell and the book Essential Cell Biology. Soon after, he worked with Dr. Lee L. Rubin at Rockefeller University in New York. Since 1983, he has been teaching and researching at the University of Colorado at Boulder. The focus of his work is the formation of mesodermal tissue, the induction of the neurocrest, and the patterns of tissue and organ formation. Another aspect of his career is the development of tools that help students to understand, evaluate, and effectively teach molecular biology to make them confident and competent and really good science teachers. He also believes that the individual needs to learn in order to recognize, work, and apply empirical data and abstract concepts in real-life situations. The result of his work is the publication of more than 140 scientific manuscripts, sites of the education science, 6 books chapter, among other contributions. Thank you, Professor. Let's talk.
Oh, goody. Okay, so I'll share my screen.
Yeah, but I'm lost, of course. Okay.
So is that visible?
Yes, I will. Okay. Thank you, Professor.
Okay. Well, thank you very much for this invitation to speak with you. I apologize right from the beginning for not presenting in Portuguese. I should have addressed this failing in my education earlier, but it came a little late. But this is really, I hope will address and get you to start thinking about it, some interesting aspects of designing courses. My main educational interest is when you think about a course, what should be taught in the course. This raises some questions that often go unanswered when people design a course. Why am I teaching what I'm teaching? And what will the students use it to explain? And this will be a recurrent theme. How will I know whether students can use it to explain a specific process? So what am I going to teach, and how do I know that students learned it? So before I begin, I'd like to thank some people who helped me through my evolution in the world of education. Kea Trujillo and Kate Henson were 2 students who did some work that I'll be mentioning here and there. Kathy Garvin-Doxis, Cedra Doxis, and Ed Servitsky worked with me to build a website called Ed's Tools that allow us to collect student data. And that was really critical for building the biology concept inventory, which is the concept test. I won't talk much about that, but it's informed a lot of my thinking. More importantly, recently has been my long-term collaboration with Melanie Cooper and her group, originally at Clemson University and now at Michigan State University. So we've done a number of projects together. For example, Sam Brzezinski has built the B-Socratic graphic system that I'll talk to you about. A large number of MSU, Michigan State students have been allow us to collect data from them, and then funding from the National Science Foundation and from the University of Colorado at Boulder. Now, what I'd like to do is, so when I was thinking about this, I was reflecting back about how we access information has changed really dramatically in the last 10 to 20 years. Nobody goes to the library anymore. You can get access to essentially every journal you want through either PubMed or the Google Scholar, and change the way people do research. Another example is that there are no user's manuals anymore. You Google it, right? You find out how to do things. So for a number of cases where you're looking for how to do something, you simply Google it. Here's a little video, which I won't show you, about how to change a light bulb. You may think you know how to do that, but nevertheless, the key advice here is don't shove your finger into the electric socket. Now, this is really good for technical issues, but it's not so great for learning how to do complex systems of knowledge. So for example, I love Sabine Hossenfelder, who's a German theoretical physicist, and she has a whole slew of videos explaining physics. She's also sort of a musician as well, so there's another set of videos on music. But here's one of her latest videos, which is Understanding Quantum Mechanics is Not So Difficult. You must be kidding me, right? So these videos presume a tremendous amount of pre-existing knowledge. For example, I, for one, have no idea what any of these symbols stand for. And it would be impossible, it's almost impossible for me to understand what it is she's saying. So when we think about how we want to think about science education, there are sort of two complementary perspectives in the world of science education. From the education side, there are questions of how it is that people actually learn, right? And there are two books on this subject. I think you can download them for free on the web, from the National Academy of Sciences, How People Learn and How People Learn Part 2. And this has a lot of really useful information about how it is that people learn. On the science side, right, there are, what is it that we should learn? Why should we learn? What we should learn and why? And there's a lot of material published on this. This is one that's very common as a framework for kindergarten to 12th grade science education in the United States. This is published by the National Research Council of the United States. Okay, so there are interesting conversations going on how people learn and what they should learn. It's probably worth and valuable to step back and consider how science has evolved, right? Originally, science was, or what we would call science or knowledge, was very practical. But when we started to think about how we viewed the world, there was often a disconnect between what we might think now as science, a sort of magical thinking. For example, anatomists, for example, would dissect animals, and they weren't doing it to understand animal structure, but they were often looking at organs to predict the future. Okay? And so you start thinking about how things have changed. We've moved from cataloging the world to developing explanatory, predictive, and increasingly mechanistic models of processes. And I think that's really important. And in that process, we've recognized the absence of vital forces and the universality of physical and chemical principles. So for example, there is no process within a living organism that contradicts physical chemical laws. So they all merge together. You can't invoke a new kind of vital force that affects the way the brain works without changing physics and chemistry. And so you have this sort of constrained system of mechanics. Science evolves also through new observations, which force new thinking. So when people started doing comparative anatomy, they started seeing things that sort of had to be explained, but weren't obviously why, and that drove the development. So these are examples of the skeletal comparisons between a mole, which is a small organism about this big, and a sea cow, which you may or may know is larger than most people by a significant amount. Here's a skeleton of a human versus a skeleton of a bird. So those increasingly accurate observations started to change the way we thought about science. So one thing that's really important in biology is to sort of recognize how it's different from other sciences. This is an interview with Ernst Mayr, a famous biologist still living. I think he's over 100 years old now. Biology is not postage stamp collecting, even though many biology textbooks may seem like vocabulary lessons where you're asked to memorize lots and lots of words. But unlike physics and chemistry, there are no real biological laws. And this is pointed out by a really good article, I think, by Beatty in 1995. And we'll talk about that a little bit more. The overarching themes governing biological systems are fairly straightforward in the generalities. And I'm not going to go through this. I published a paper on this a while ago, thinking about the conceptual foundation. But there are evolutionary processes, there are molecular foundations, and there are network behaviors. So if we think about a biological system, we're always thinking about the interactions of objects, either at the cellular level, the molecular level, the ecological level. There are sort of versions of these network behaviors. So you think about how biology is different. And this is from another article by Ernst Mayr, Cause and Effect. There are really sort of two kinds and really distinctive kinds of explanations. There are proximal causes, which is how things happen, how molecules interact with each other, how genes are turned on, how the proteins in a cell interact to produce various behaviors. And this applies at the molecular, the cellular, the organismic, and the ecological level. And these are all consistent with physical chemical principles. There are the ultimate causes, which is why things are the way they are. This is the product of Earth's evolutionary history, and it's trickier to know because you weren't there. Many of these processes were established millions and hundreds of millions and maybe billions of years ago. And the context in which they were established is impossible to know, but it's worth recognizing that life has been evolving for a very long time. It's been subject to various accidental traumas, as witnessed the collision of the Earth with a meteor about 65 million years ago, which appears to have led to the extinction of the dinosaurs. We do not know what would happen if we could rewind the clock of evolutionary time and run it again. Would it produce human beings? Would it not? We don't know. And in fact, it's unknowable. So, but it's worth realizing that the way things are the result of evolutionary processes. And so these are two different levels of explanation. So biology has really moved from a descriptive to a predictive and mechanistic terms, but many teaching materials have remained at this pre-mechanistic stage. And we're often asked to memorize things without knowing. And I say that my premise, the theme I'm talking about here is that the descriptive details included in a course or a textbook need to be justified to the student, because the student, it's very important for the student to understand why they're being taught what they're taught, how it's useful to them, what they can do with it. So they need to be justified based on whether that it helps students generate explanatory models of phenomena. And I'll give you some examples of that, okay? So here's what we're going to talk about. So an important example, and this is a point raised by Bruce Alberts, who is another author of the textbook Molecular Biology of the Cell with Martin Raff, is the cell is a collection of protein machines. So when we're thinking about what's going on in the cell, everything is a protein machine, essentially, ranging from how energy is captured, how molecules are synthesized, how genes are expressed. And we've got examples of that. Here's an example that I particularly like. This is a DNA importer. This is a system of bacteria, which is turned on or off. It's actually regulated by a process called quorum sensing, because there's no reason to import DNA to build this complicated protein machine unless there's DNA in the environment. So if there's DNA in the environment, you realize it because you recognize that there are other cells like you around, other organisms like you. You turn on the genes that encode this, and this machine grabs DNA from the external media. It imports it in through the membrane of the bacteria. It turns it into a single-stranded piece of DNA. It degrades part of the strand. And that DNA, as it's imported, can either become nucleotides or food, or it can be the information in that DNA can be incorporated into the importing cell. This is one of the mechanisms behind horizontal gene transfer, the way genes can move within populations of microbes. It's worth I think it's worth thinking about this from the machine point of view, because this is the basis of what's called nanopore DNA sequencing, which also uses a pore in which a single strand of DNA moves through. Because the bases of DNA are different sizes, as they move through the pore, they obstruct the movement of ions, and you can read the sequence of the DNA as it moves through this pore by reading the current flow through the channel. So this is a molecular machine that's used all the time. You can buy it if you want. There's a video that I won't show here, but I think it's worth realizing, that essentially everything going on in the cell at the cellular level is a molecular machine. So let's think about what do I, as a student, need to know when I think to explain the behaviors of a molecular machine. So I have to know how molecules interact, right? What determines the specificity of interactions, the stability of those interactions, okay? And that's basically coming from chemistry, essentially. I have to know how molecular machines do work, right? So, and that's driven by coupled chemical reactions. I'm going to take favorable chemical reactions and I'm going to use them to drive unfavorable reactions. So for example, the synthesis of DNA is an unfavorable chemical reaction. And so to do that, I have to couple it to a favorable reaction. So I have to understand that coupling of reactions, chemical reactions, what makes a chemical reaction favorable or not favorable. And how do changes in protein shape influence their interactions with other molecules and their function? So I have to get an idea of what determines protein shape and all those processes. So this is results from a study with Melanie Cooper's group. So do students really understand molecular interactions? In this case, what we use is the B-Socratic system, which I'll talk about more a little bit. We asked students to write down how hydrogen bonding works, and we got examples of student behavior, and you can see that there's a wide range of drawings made and descriptions given. Some are fairly simple. This person is a little confused. They're confusing a covalent bond involving hydrogen with a hydrogen bond. This is more correctly drawn. We can do that for hydrogen bonding, dipole-dipole interactions, and London dispersion forces, and trying to understand that. So it turns out if we look at how many students in the class are drawing that correctly, we begin to understand what's going on. And we've published this paper some time ago. But what's interesting is in a traditional curriculum, the idea that hydrogen bonds and dipole-dipole interactions and London dispersion forces occur within a molecule, and we're talking about small molecules, is persistent, all right? And so you may not have even suspected that students think this way, but if you don't recognize it, you're going to have trouble getting them to understand how molecules or proteins or other kinds of molecules interact with one another. Not having an accurate understanding of such a core concept makes developing a working understanding of core biological processes difficult, if not impossible, right? So processes such as DNA replication, gene expression, and the effects of mutations become mysterious rather than predictable. So let's think about it. What do I need to know about genes? So you can reflect on this about how you were taught. So there's a basic definition of gene, which is it's a physical object that occurs in various forms and it influences phenotype. So what do I mean by that? Well, it's a region of DNA, and you could talk about how that was discovered. The various forms are alleles. Okay, so various forms of a gene are alleles, and that there are multiple alleles within a population. And phenotype really is just an organism's traits, something you can measure that is different between different organisms, and that impact, and from an evolutionary perspective, that impact reproductive success, and that leads to evolution. So this is the level that you need to understand it from the point of view of just basically evolutionary behaviors. So a more important question, and that, so you will, I hope it's true, but most people are not taught a gene in that way. They're taught as it in parts, okay? So there are many parts or potential parts to a gene, right? And so you're given a lot of things to understand. This is a question, and if there's a question, somebody should interrupt me. So this is taken from a website that 10 students answered this question. Now the real thing you should be asking yourself is, why am I being asked to learn this? And the reason is it makes possible plausible predictions for how changes in gene sequence might influence gene expression and the gene product. So a mutation that occurs or a variation that occurs here, or in the intron here, or in the promoter here, or outside the coding region, transcribed region here, we can make predictions about what effect that will have. There's a lot to memorize here, and it doesn't really mean anything unless you're going to use it for something. The same thing goes about protein structure. So often students would be introduced to, here's the sequence, the primary sequence of a protein, the secondary structure of the protein, which is the way it forms alpha helices and beta sheets, the tertiary structure, which is how it folds in three-dimensional space, and the quaternary structure, which is how it interacts with other polypeptides to form a functional protein. Once again, memorizing this on its own has no value unless you're asked to use that information to make plausible predictions about how changes in an amino acid sequence might in fact influence structure, activity, or behavior. So where a mutation occurs in the structure will have that effect. Again, there's lots to memorize unless you need to know it to use it for something. So what's important? That they memorize the names of amino acids. I've had a number of students in my department tell me that they've been asked in various classes to memorize the names of amino acids multiple times in their classes. Now, is that worth knowing? I do molecular biology on a regular basis. I don't remember all the names. I look them up. But what I can do is deduce the chemical properties from their structure. I can look at an amino acid and tell you how it will change the ability to interact with water or other molecules. And that's the important key here. Now, in another study, that ability to read structure, read physical properties or deduce physical properties from structure can be very difficult. So there's a paper, Lost in Lewis Structure, which examines whether students can deduce the physical properties of a molecule by simply looking at its structure. And in fact, it's pretty rare, right? So you need to focus your instruction on saying, what is important about this here? Can I use it? Because depending on what amino acid is substituted for which one will determine your prediction of the effect on protein structure and function. All right, so, and one more before we go on, to understand signaling responses. So I've just recently been teaching developmental biology, and a key to many biological processes, from quorum sensing in bacteria to pattern formation during embryogenesis, is based on cells' responses to signaling. And that can generally be as a sigmoidal function. So part of the question now becomes, well, why doesn't the process, why don't you get a response until you reach some initiation point, and why does the response saturate? And the sort of limit example of this is a threshold response. This is used many places in biological systems. At a certain level of input, there's no response. At a slightly higher level of input, there's a full response. And this is part of how embryos are patterned, how cells respond to signals. And so you think, well, what do I need to know? I need to understand, well, why am I not getting a response here? There are reversal mechanisms going on. So unless I have a large enough signal, I'm not getting enough to produce the outcome. There's molecular turnover, interaction dynamics. molecules may be coming together and going apart really quickly. There are feedback interactions that can influence the shape of these kinds of curves. When we look at thinking from earlier courses, we ask students, in terms of an increasing concentration of transcription factor, what do you predict the response gene level will be? And you can see that the students produce a range of responses, some of which are close to what we expect, others make little or no sense or are not explained. So coming back, we want to ask the question, have students been given the tools needed to explain responses, or how we get a threshold response? Could they possibly make a model from what they've been taught about how I would produce this kind of response and how I would describe it? So this leads to the question, how do we know what students know? And this is a key question in education. I would argue that the gold standard is free-ranging Socratic conversation in which the assumptions of the student are revealed, justified, and their skills are displayed to an informed partner, conversational partner. This clearly is often not feasible, so there are various levels of compromises. There are concept tests, there are various forms of multiple choice and short answer exam questions. The real problem is that there are limitations to these kind of questions. They often fail to reveal the level of understanding and often deal with remembering or recognizing terms rather than applying knowledge. Okay, and I won't talk to you about efforts we've made in this area about building concept tests, but suffice it to say that even concept tests, which are more rigorously research-based objects, are in themselves limited. So we're looking for more revealing assessments, and this describes a project that started in Melanie Cooper's lab and was really driven by Sam Brzezinski, a graduate student in computer science. And this idea is that we can understand what students are thinking by letting them draw for us, as well as explain in words. So here is a classic example in physics. Here's a diagram of a rock attached to a rope, to a person who's sitting at the center, who's spinning it around their head, right? This is sort of a David and Goliath sort of model. And so I say, well, draw what happens to the rock if I let go of the rope. And this has the advantage of showing us the range of responses that students are producing and provides a context by which we can start to address, well, why is a person drawing it going this way versus a person drawing it going that way? What are they thinking? And equally important, we look at students' descriptions of the ideas involved. And so this has been very valuable to us. It's available. Part of the problem is, I'll just say, we generally make educational materials so that they're free and easy to access. The problem with computer software and web software is that it's expensive to maintain. So we're still in the process. This is available online, but we're still in the process of figuring out how to keep it funded. Currently, students at Michigan State are paying for it as part of their chemistry course, where they get a free textbook, but we're still working on that. Our updated solution is to incorporate these kinds of B-Socratic activities in various curricular materials. So one is Chemistry, Life, the Universe, and Everything, which is a general chemistry curriculum. That material is free. And then Biofundamentals, which is intramolecular biology. And you're free to take a look at those. I'll make these slides available whenever, but the acronyms are pretty good. If you Google them, you'll find it. Most recently, we finished Organic Chemistry, Life, the Universe, and Everything, and a transformed organic chemistry curriculum, which is also free and available. Now, what makes these, particularly the chemistry resources, really good is because Melanie Cooper has been in a unique position to be able to do comparisons between traditional courses and CLU, both taught in a more interactive way, not simple lecture. And so we have a fair amount of data on their efficacy. What I thought I'd do is I'd tell you a little bit about what was the catalyst for CLU. And we had a conversation, Melanie and I, at the Chemistry Education Gordon Conference some time ago, a decade ago, on the energetics of bond breaking and bond formation. So as you well know, when a bond is formed, that bonded molecule is more stable than the unbonded form. And so energy is released. And we were curious about the fact that when two molecules came together to form a stable complex, you never saw where the energy went. And a very talented collaborator, Tom Lundy, built a whole slew of these in Flash, which is now obsolete, that allowed you to look at the behavior of molecules. But that idea that molecules are bonded or of lower energy than molecules that are separate, that it takes energy to knock the two molecules apart, got us thinking about how concepts were being taught in general chemistry and in biology. The CLU curriculum is focused on chemistry, and it's focused around the structure and properties of molecules, the energetics of molecule formation and interaction with solvent, how bonding interactions, which are relevant in that area, and change in stability. Now, what's interesting is I showed you this before. This is students thinking about hydrogen bonding, dipole-dipole interactions, and London dispersion forces. Generally, they believe that they're occurring within molecules in a traditional curriculum. In the CLU curriculum, we have them becoming more and more aware that they occur between molecules in these contexts. And in studies I won't show you, if you look at the impact of this curriculum as you go, say, through organic chemistry, you see these presumptions continuing. So students essentially never lose the idea that these intermolecular interactions are occurring within a molecule, even as they go through an organic chemistry course. Now, why does it matter? So Melanie and her colleagues at Michigan State have taken a look at the impact of transitioning to more functionally, conceptually centered curriculum like CLU. And what we see is, this is the grade point average of students. And we see an increase in grade point average, and these are very large classes. These are hundreds of student classes of about a half grade point. And I'm not sure how that translates into your experience in Brazil, but it turns out that this can be the difference between having to in the United States, having to take the course over again. And many times when a student is asked to take the course over again, they leave. They leave the science curriculum in between moving forward. And there's just been an interesting paper from Scott Freeman, Harris et al., his group at Washington, University of Washington, that suggests those who just clear this hurdle, who pass the course and don't have to take it again, they succeed in their subsequent studies as well or better than those who score higher. So this is an increase that makes a difference in the lives of students going through the curriculum. Okay, so I just put this here for your, you can find various examples of data about what we've learned what we in the Melanie Cooper's group have learned from studying the impact of using CLU in various contexts, and you can Google Melanie's name to get more recent work. So sort of to sum up, what I would like you to think about when constructing a course is that you consider why I'm teaching a particular topic, the detail, and even if you're in a position where you're constrained by your curriculum, so somebody's already told you what you have to teach, providing the context so that the student understands why I'm being taught a particular topic, and particularly, will I use it again? What will I use it to explain? Why does it matter that I understand it, right? So for example, if I want to start making models of how a mutation or an allelic difference or something, and particularly in the world of COVID, what would happen if I change this amino acid in, a spike protein to that amino acid in a protein? Now I can start, as I understand it, I can start making plausible predictions. They may or may not be right, but they make sense. And then how will I know whether students recognize and can imply that information correctly, figure out ways of getting them to show you whether they're understanding it. And I would just say that you should feel free to contact me. All these educational materials I mentioned are freely available. I have an occasional blog, which I don't write that often. But so I'll end here. Thank you for your attention, and I'm happy to take essentially whatever questions you may have. And I can stop sharing my screen. So I'm happy to entertain questions.
Hi, Professor. It's a great
Hello.
talk. And someone has a question, because I have a lot of questions. And you were talking about to teach things like the mechanic of proteins, amino acids. Sometimes I think how it's possible to connect the idea today of moonlighting proteins, that protons that have a non-canonical function that we are used to work every day, to teach every day. And now, for example, when you were talking, I was wondering a lot of things, because we have to select your point of your class. What you teach depends on your class, depends on your student, depends on the background of these students, and it's a huge problem, because, for example, sometimes you have to match mathematics with chemistry and the idea of biology, and we have so many information today. Right. And it's complicated. For me, it's complicated. Sure. I have to face in this challenge for my future as a teacher.
No, it's extremely challenging, right? Because the reality is, and you know, I love these weird molecular mechanisms, and I love the weird things that you never expected to occur. I think what you're trying to get across, and you should be happy, is if students can make a plausible mechanism. It doesn't matter whether it's correct, because one of the more interesting things about biology and how it makes it different is that essentially no one is smart enough to predict biological systems, right? Whereas physicists think they're smart enough to predict the structure of the universe. Nobody is smart enough to predict a biological system, because there could be another protein doing some other thing that you didn't know about. I've spent a fair amount of time studying proteins like plakoglobin, beta-catenin, and chibi. These are proteins that have structural roles in the cell. They also go to the nucleus and regulate gene expression. So whenever you think you know what a protein does, you're almost certainly wrong. It probably interacts with 4 other proteins. It probably does 3 other systems. You want to back away from the idea that I'm looking for you can only memorize the quote unquote correct answer. There's no way to know it. There's no way to deduce it, but you should be able to make a plausible model. If somebody gives you the components, you should be able to start putting together predictions. And what's important is to get students to not feel like they're a failure because they don't know the right one, because it's not possible to know the right one. You just memorize the right one, right? But the ability to say, how is it that I get a sigmoidal curve, right? How is it that, why does it saturate? So think about those processes that way. I mean, there are so many complications. I would normally put up, there was a cartoonist about 50 years ago, Rube Goldberg, would make these machines where, it was like, how do you lift a cup of coffee, and it would have 47 different components in it that went from pushing a button to having a coffee thing. because biological mechanisms are the product of evolutionary processes, they are not intelligently designed. They work, but they are not intelligently designed. And so you want to give people the idea, okay, so if molecules interact, they have to have surfaces that are complementary to each other, and charged and uncharged groups of various types. So you can make predictions. And part of it is saying, can you give me a mechanistic model based on a coherent understanding of intermolecular forces or whatever, and don't worry whether it's right? I mean, that it's correct, okay? That it works is more important. Now, this is a tricky problem because you look at many biological systems, they have even, one I've been thinking about a lot is a very simple gene network. Well, the outcome of that gene network depends on assumptions about, affinities between molecules and all that stuff. You can't predict unless you make a mathematical model and measure all the affinities and half-lives and all this other stuff. You can make plausible predictions and then explain why you said what you did and what you think it should look like. So I think the key is to not let the detail of biological systems overwhelm you, because it's overwhelming. There's a reason that it's not trivial to make a drug. Okay? There's a reason it costs billions of dollars to make a drug that treats a disease, because you can't just look at it and say, oh, like that.
So it's not the amount of knowledge, but the quality of knowledge.
It's the ability to use it. It's ability to recognize the underlying principles, okay? So you say, if I look at, say, the ATP synthase, which allows hydrogen ions to go through a membrane, turning this molecular machine and driving the, essentially catalyzing the synthesis of ATP from ADP, is it worth memorizing the 20 or 30 subunit proteins of that structure? Or is it worth understanding that there's a gradient and a catalyst and a coupled reaction?
Yes, because the idea that I have is that when you are in research, you always focus on your own research. And sometimes it's because easier to understand the root or some phenomenon, because you're always reading about this point, but you forget the other points. When you have to contextualize your research and your findings, you have to contextualize and transfer for the society.
Exactly. You have to recognize the difference between working out the details of a molecular mechanism, right? Particularly in the new age of molecular biology, where I could look at single cell RNA sequencing and essentially determine every gene that's being expressed in that cell and how those genes change, in which there are hundreds of changes going on, no matter, so like if you look at a cell, you can change hundreds of genes. And then there's beautiful work looking at, here's a cell type, I perturb it with a turning on some genes or other genes, and I get many, many different behaviors because the system is stochastic. It has lots of different things going on. If I look at every protein, and that is determined experimentally, but it's not central for a student's understanding. That's an example of, I'm focused on this machine. That same machine may be different in a related organism, right? So it's not even transferable from species to species necessarily, right? That's one of those interesting things you realize, like the way a mouse embryo develops and the way a human embryo develops have important, significant differences between them. Right? There's a reason that drugs in the mouse don't always work in humans.
Right. Can I ask a quick question?
Yeah.
Now, do you think that, I think in terms of what does a student need to know to understand how a cell works? So I think that a cell in some way is a simpler system, and there's lots of, still very complicated, but you can define the range of knowledge if you try to understand what is essential knowledge to understand how the cell works. Of course, the animals have very different solutions, but a cell has sort of very few solutions. I mean, we know that life can be, probably can have in different coding process, can be RNA or DNA, but now on Earth, we only have DNA-based life, and
right.
Somewhat simple. So I think that, so the question would be, you think that cell biology is some way easier to define the contents that should be taught, considering it's a very standard situation?
I'll go back to what I said before. It all depends on what you want the student to do. Okay,
yeah.
So the cell. I mean, so I don't think ecologies are that much more complicated, right? They're objects that interact. What's a cell? Cells have objects that interact. In an organism, cells are interacting. They're sending signals to one another. They're responding in various ways. I don't see them as significantly different from one another. Now, the question would be, so you're doing a cell. You say, okay, it's a eukaryotic cell. It has multiple compartments. How do proteins get where they're supposed to go? Well, they have to, so there must be a signal that must have a receptor that says, okay, here I go there. How do I say, here's the nucleus? Okay, so there's a signal that says this is the nucleus. There's a machine that imports. So you're talking about, okay, what do I want the student to be able to do? Do you want them to be able to predict where a newly synthesized protein in the cell will go? Then you start talking about nuclear localization and export sequence. Maybe then you really care about whether you talk about the machines at the nuclear pore that are determining whether you're going in and out, right? But it's all just, okay, there's a machine. Now so I would say an important idea in biology is that life never stops. Life never starts. It's been running continuously for about 3.5 billion years. So everything is occurring in the context of a functioning system, okay? So the question is, well, what are the characteristics of that system? In a eukaryotic cell, there's a topology that's pre-existing. So you're already talking about, well, where am I going? How do I mark it? And you talk about it because you care. How does a protein get synthesized and secreted? Or how does a protein get synthesized and go to the nucleus, right? How does a protein fold? I'm very much into chaperones at the moment, right? And the idea that people want to do folding of proteins in the test tube. Well, proteins don't fold in a test tube. They fold in the context of many molecular chaperones that are doing that process. So I would, so again, I'll just come back to the same idea. What do you want the student to do with it, right? I mean, I've run into the Krebs cycle so many times, and I've never seen anybody ask a student to do anything with it. So it makes no sense to memorize it. It's just a series of coupled chemical reactions, okay? Mm-hmm. Now, if you want them to use components, which you could in a research project or in a particular biological behavior, you could say, well, this component of the Krebs cycle is also a regulator of this thing, right? So now you're talking about, well, but you now have a reason why you think it's important to know it. You're telling them what they can use it for. And you yourself are thinking about what I can use it for. Okay. What would a mutation, what do I need to know to be, so your point is really good. Well, if I want to understand plausible mechanisms of mutation, I have to know that proteins can go to various places in the cell. If I mutate the signal that controls nuclear localization, a transcription factor could be made perfectly well and not function. Mm-hmm. Okay. So again, it's the same theme. Why do I need it? What am I using it for?
Okay. So Miria, can we move on?
Yes. Any other question? So, can you turn to Professor Mermelstein? Professor Klymkovsky, now I'm sure about your name.
No, you're an expert.
I really appreciate it. your talk. And thank you very much for sharing your experience with us. I will invite Dr. Cláudia Mermelstein to speak with us to her presentation. And Cláudia, please open your camera. While she's opening her camera, I will talking about Professor Cláudia. She's full professor from Biochemical Science Institute at Federal University of Rio de Janeiro. She obtained her PhD degree in 1992 under supervision of Dr. Moraneto and Dr. Howard Holzer from University of Pennsylvania. During her PhD, she worked with embryonic Chuck myogenesis. She's vice coordinator of Morphological Science graduate program. Her work is focused on signaling routes associated with the control of differentiation and skeletal muscle. Dr. Mermelstein also coordinates projects in cell migration cancer and has interest in cell biology education. She teaches cell biology for undergraduate courses, such as medicine, pharmacy, and biomedicine since 1993. Cláudia?
Yes. Can you see my presentation?
Yes.
Yeah, okay. So thank you very much, Miriam. Good evening to all. So it's a pleasure for me to talk today about the identification of main topics in cell biology in Brazilian universities. Just one second. Okay. Before I begin telling my work, I would like to talk a little bit about my background and the motivation for this study. I did my undergraduate studies in biology, in genetics and ecology at the Federal University of Rio de Janeiro. And then I did my master degree in biophysics with Professor Vivaldo Moraneto and Professor Carlos Chagas Filho, studying the cytoskeleton of electric eel cells, the electrocytes. And then I did my PhD studies in biophysics, both at the Federal University of Rio de Janeiro and at the University of Pennsylvania, working with Dr. Howard Holzer, studying the role of actin-associated proteins in muscle cells. 2 years after I finished my PhD, I was hired as a professor at the Biomedical Science Institute at our university. And then I began doing research in embryonic chick myogenesis and also teaching cell biology for undergraduate and graduate students. So I'm teaching cell biology for the last 26 years. So during this period, many questions came to my mind, such as some similar to the ones that Dr. Klymkovsky just told us. Some questions were, which topics of cell biology should we teach undergraduate and graduate students? At what level of complexity should we teach cell biology? And which teaching methodologies are the best for cell biology? So I will talk a little bit about each one of those questions today. But before I continue, I will just talk a little bit about what is cell biology, since today we have here many biologists, but also we have many non-biologist audience. And cell biology is the study of the structure and function of cells. And how can we teach cell biology? In my way of seeing this question, we have at least 6 different approaches for teaching cell biology, and they are not exclusive. The ideal situation for me is to combine all of them. The first one is to describe the cellular structures, cellular organelles, or even structures, complex structures. So 2 examples will be the cell cytoskeleton is composed of 3 systems of filaments. This is a descriptive way of showing cell biology, but in some slides I will show you the importance of that. A second example will be the cell membrane is a phospholipid bilayer. This is just a description of cellular structure. The second approach is more a dynamic view of the cell. Since the cell have many processes going on, it's very important to explain to the students how these processes can occur, such as cell division, cell death, cell migration, cell adhesion, and cell differentiation. The third approach that I use a lot is to show the students the historic view of the discoveries of the cell biology field. We can show the students the first paper that appeared in a specific topic of cell biology, and the students can follow how this specific topic evolved, how it changes from the early papers to papers that were published like this year. So this historic view is very important for the students to know how the scientific process occurs. Another important approach is the evolutionary view of cell biology. As Dobzhansky said, nothing in biology makes sense except in the light of evolution. So evolution is very important. And I use evolution in all my classes, telling the students about how proteins evolve, how cells evolve, how organisms evolve, how organelles evolve, and so on. I also like to talk about the scientific process in all my classes, how every paper, they have a question, they have a research, they have a hypothesis, experiments, and they have observations and results, conclusions. So this is very important to the students to know how science works, and they need to be very critical about this. The sixth approach is an experimental view of cell biology. This is more like a practical approach, is to take the students to the lab and show them how cell biology techniques work, how they function, and the students can try to repeat some of these techniques in the lab, such as optical and electron microscopy, cell culture, cell transfection, electrophoresis, and so on. So they can see the theoretical part in lectures, and they can also see how just one second. They can also see how the techniques of cell biology function. Another point that I think that's very important is to use this spiral educational learning. So the students need to revisit cell biology concepts in different moments of the discipline. And I think that helps the student to gain learning skills. For example, if you at day one, you can give a lecture to the students telling that microtubules that are part of the cytoskeleton participate in the dynamic processes in the cells. And then at day 5, the students can present a seminar showing how microtubules participate in cell division, which is a very dynamic process. And maybe at day 10, you can bring the students to your lab and show them using immunofluorescence microscopy how microtubules participate in cell division. So you can show them that this is a very dynamic process in the cell. So in each different day, the students will see the same topic, but in different levels and in different kind of classes, and they can gain knowledge by seeing the same point of the same point in cell biology by different methods. Something that I think that's very useful also is to use schemes and diagrams and models and drawings. I do a lot of schemes during my cell biology classes. Even today, I did some drawings. I like to draw the mitochondria, the plasma membrane, and ask the students to do these drawings. When the students try to do the drawing, you can see whether they are understanding or not how this organelle or how this specific process is working. For example, how the mitochondria can produce ATP even in this complex organelle if they don't have these two different compartments. So the students need to understand the structure of the mitochondria using this scheme and how this scheme is related to the function of the organelle. The same thing is applicable to the plasma membrane. The students need to understand how they can draw a bilayer, a phospholipid bilayer. They need to understand that there is a hydrophobic tail, there is a hydrophilic head, and if they don't understand that, it's impossible to understand how the plasma membrane will function as a barrier. So I also use many diverse media, such as videos, video microscopy of live cells, and schemes together with the videos. Since cell biology is shows a very active part of the cell, how cell functions, it's very important to show them videos, like a living cell migrating, as it's shown here in this movie. And in the top, we can see that a migrating cell has many different filaments from the cytoskeleton within the cell. So here's a cell that was injected with fluorescently labeled proteins, and you can compare the whole cell with the cytoskeleton working in the same process, and at the same time, you can show them schemes of how these proteins from the cytoskeleton can polymerize and depolymerize, and how this relates to the cell migration that we are seeing in these movies. So together with all those questions, we made some meetings in our undergraduate program of cell and developmental biology at our university. And we made 2 lists of the cell biology topics that we consider essential, like in these 8 topics. It's a very short list. And we made also a large and complete list of cell biology. Why we made these 2 lists? Because we have some disciplines that are very short. They have like 8 classes of cell biology. They are usually an introduction to another class of histology or embryology or biochemistry. So cell biology is sometimes we have little time to talk about so many things. So we have now an essential list with only 8 topics. And when we have time, we have these 25 topics in a very complete list. We have methods in cell biology and all the organelles, all the cellular processes to teach the students. So, but we need to understand that each topic that I'm showing you here, like plasma membrane, it's not enough to decide that plasma membrane is a very essential cell biology topic. We need to discuss what we should teach in a plasma membrane class for undergraduate or graduate students. So we made a discipline in our graduate program where the students participated in trying to design what is essential to be taught to students in a class related to plasma membrane. So here you can see that there is membrane structure, history of the discovery of plasma membrane, functions, and so on. So we did this graduate class for all those topics that I'm showing you here, but I don't have time today to show you all of those. And we then decided to go further in this study, which I did with Manoel, and we decided to analyze what's being used in Brazilian public universities, what are the teachers using in terms of topics of cell biology when they teach for undergraduate students. So we decided to make this analysis with 2 main goals. The first one was to analyze the differences, the possible differences between different courses. Since in Brazil, we have different courses like medicine, biology, pharmacy, and physiotherapy from the beginning. Since the students get into the university, they have already a separated course for each one of these careers. So we analyzed 13 courses. Here I have only 4 listed, but the complete list is with 13 courses. We also analyzed the differences between universities. We analyzed a total of 15 federal universities in Brazil. I will show you in a few slides. So we did this analysis of cell biology content being used in Brazilian public universities. And we also did, for comparison, an analysis of the 3 most used textbooks in cell biology: The Cell from Alberts and colleagues, Molecular and Cellular Biology from Lattes et al., and Cell from Cooper. We decided to analyze the content of this book and to compare it with the Brazilian contents used in the universities. So the results we published 3 years ago in this paper, Analysis of Undergraduate Cell Biology Contents in Brazilian Public Universities. Was really interesting to do this work. Was a lot of troubles and problems, but at the end was really fun. Here is one example of a cell biology discipline content syllabi, and I use here one of my own disciplines. So here we have everything that we needed for our studies. We have the name of the topic of cell biology that the discipline was using. We have the duration in hours of each class. We have the name of the discipline and the name of the course, here pharmacy. So we decided to use this kind of document for our analysis. And so we decided to get this information from the internet, because in Brazil we have a law saying that the content of all disciplines from federal universities must be posted in the internet. But this is the law. It's very different from reality. So in fact, we couldn't get these contents from the internet. We got maybe 20% of our data from the internet, and 80% I needed to send an email to each individual cell biology professor from all over Brazil and ask them to send me their contents like this one. So by email, I got many answers. So at the end, we had these 15 universities, and we have these 13 courses. It's important to tell you that some of the courses were unique to some universities. So for example, aquaculture and oceanography, they were not present in all those universities. They were present only in 2 of those universities. So at the end, we have received 150 different cell biology disciplines, okay, to work with. So we have represented Northeast Brazil, Southeast Brazil, and South Brazil. We could not get any content related to cell biology from North Brazil. Maybe in the future we can extend this work. So we collected all those data that we received, and we made a giant worksheet that we call that the full collection of our results. And we have the first column, we have the name of the university. You see that the colors change as the university change. In the second column, we have the course name, like pharmacy, medicine, biology, and so on. Sorry, Klymkovsky, it's all in Portuguese. It's the only slide that is in Portuguese, because this is our main full collection of data. So it will be really hard to translate everything. But I did translate some of the topics. So here we have the name of the discipline. You will see that varies a lot. And here is the duration in hours of each discipline. And here in yellow is the really interesting stuff. We had 28 different cell biology topics that were present in these Brazilian documents that I received. So here I have few of them. The worksheet is larger than the one I'm showing here. So here we have evolution of cells, molecules, techniques, plasma membrane, cytoplasm, adhesion, and so on. So we analyzed all the data from the worksheet, and here I listed all the 28 topics obtained from this information, these documents that the professors sent me. And you can see that we have evolution of cells, nuclear structure, and so on. We have more than we could imagine of topics present in those documents. We have some topics that we were really surprised to see, like cytoplasm. I don't know what we can teach in 2 hours of a usual class and talk about cytoplasm, but that's what we found. So we used all this data to analyze in a single graph. So in each blue bar, you have one of those 28 topics of cell biology. And at the top of each column, we have a number that's the percentage in which this topic was found considering all the 150 different cell biology disciplines. So it means that 50% of the disciplines teach evolution of the cells. And for example, we have cell signaling was present only in 32 of those disciplines. In the red arrows point to the 4 topics that were present in the highest frequency: plasma membrane, cytoskeletal, nuclear structure, and the plasmic reticulum. They were followed by Golgi and cell cycle and mitochondria. So we could see that some topics were really well represented, but other topics were really in a low frequency. And so here's a summary of what we found in the first part of this paper. We found, as I just told you, that the most frequent cell biology topics were plasma membrane, cytoskeleton, endoplasmic reticulum, nucleus, Golgi, and mitochondria. The less frequent cell biology topics were cell signaling, cell death, cell differentiation, endocytosis, and regulation of gene expression. Curiously, the first one, the most frequent, could be understood as organelles description. And the second one that were really have a low frequency could be understood as cellular processes. So this is something that we were really worried about, me and Manoel, and its conclusion is written here. Brazilian cell biology disciplines contains more topics related to the description of organelles, structures, than dynamic cellular processes. And here I put what we listed as structures and processes for comparison. Something that also called our attention was that we saw some unexpected topics, such as cytoplasms, as I told you, but also virus, cell metabolism, bioengineering, with sometimes very low frequency, but we were surprised to see some of those topics instead of cell signaling, for example. So I will talk about that in a minute. Something else that we did was to analyze the duration of each discipline in hours. So the average duration of a discipline, of a class, for a discipline as a whole, was 60 hours per semester. But we found some disciplines that had only 15 hours and others that had 150 hours. So there was a huge difference in the duration of cell biology disciplines. And we know that the duration do not tell us a lot, but it's something. We have more time to teach cell biology, or we have less time to teach cell biology. So this is something that we can discuss later. We also analyzed the name of the disciplines, and this is listed here in this table. And the name of the discipline tells us a lot about the history of cell biology in Brazil. We can see that we have many different names, and usually cell biology is not alone. It's together with embryology, genetics, histology, medicine, and so on, and biochemistry also we saw. So it means that in Brazil, we know now for history that cell biology is usually used as an introduction for another discipline. So cell biology is used here as an introduction for embryology, an introduction for histology, and so on. And we also found the name cytology, which is an old name, a more descriptive name, and this changed over time to cell biology. So this is shown again in this slide. So cell biology is a relatively new area of research in Brazil when we compare with other countries. The Brazilian Society for Cell Biology was created in 1978 by Professor Junqueira. And what we can understand today is that cell biology was an introduction for all those different disciplines. But as the time passed, cell biology gained a lot of strength, a lot of data, and now some of these disciplines are beginning to change their name for only cell biology or molecular biology of the cell. So this is changing over time. And the interaction between cell biology with those different disciplines is very important for cell biology. We have many different proximities, and those areas help to change and to evolve together with cell biology. So the analysis of the textbooks that I told you, that was the second goal, we did in the following way. We analyzed each book in relation to the index of the book. So for the cell from Alberts, we found that there was 21 topics, and we counted the number of pages dedicated to each topic. So we could do a percentage for each topic. We did the same for molecular biology of the cell, for the Cooper book. And the first thing that we did was to find what was in common, what those three books had in common. And then we made this list of 15 different topics that all those three books had in common. And then we also analyzed a Brazilian book from Professor Junqueira and Professor Carneiro. This was the first cell biology book published in Brazil. We analyzed an early edition and one late edition. And the early edition, when we compare it to the later edition, it's very interesting to see how the index changed. For example, there was no cytoskeletal in the first edition, and you can see now the cytoskeletal word in the later edition. The same is for all those green words. All green words means that those are some new words that appeared in this 9th edition. So cell signaling appeared, cytoskeletal, cell cycle, many different cellular processes gained space, and some only structural organelles became lower in frequency. So that was very interesting to do this analysis. And then we came up with this graph, this representation, in which we can show the textbook from Alberts, the cell, counting the pages dedicated to each one of the chapters. We found that those are the chapters or the topics that have a most abundant, most frequent words in the book. So cell differentiation represents 13% of the book, regulation of gene expression 12%, biomolecules 12%, methods in cell biology 9%, cytoskeletal 7%, and cell signaling 6%, and so on. So we decided to compare the percentage of these topics present in cell biology book in the cell with the results that we collected from the Brazilian universities. And then we saw a very high discrepancy, very high differences. For example, biomolecules were had a very low frequency in Brazilian classes. The same for cell signaling, only 32%, cell differentiation and regulation of gene expression. Those 4 red arrows I pointed here just to compare. Those are the ones that are most have a high frequency in the book and have a very low frequency in our Brazilian universities. So this is something that we are discussing together with the Brazilian Society for Cell Biology and professors of cell biology, try to understand why such topics such as cell signaling and the regulation of gene expression that are so important for a cell biology student, why they are so rarely present in those disciplines. So I just put this here to understand, it's very important to understand cell signaling in any cell context. At least you need to teach the students the general aspects of cell signaling. You don't need to give all the names and all the different signaling pathways, but you need to teach something related to cell signaling, cell differentiation, and regulation of gene expression. In my point of view, those are essential topics. So with that in mind, we came up, me and Manoel, with this list of the 14 topics that we think that should be present in any undergraduate students discipline for biological or biomedical students and courses. And those are evolution in cell biology, methods in cell biology, membranes, cytoskeletal and cell motility, cell adhesion, and so on. I will not read everything. So we published that. And how we made this table? We made this table based on those 5 topics listed here. We consulted the cell biology learning framework from the American Society of Cell Biology that's shown here. We consulted also the 3 textbooks, cell biology textbooks, the Cell from Albers, from Lattes, and from Cooper, and especially looking at those topics that were in common about those 3 books. We also took in account the meetings that we have with our undergraduate program of cell and developmental biology, meetings we have with members of the Brazilian Society of Cell Biology, and of course, our long experience teaching cell biology from different courses, medicine, biomedicine, pharmacy, nutrition, and so on, that we teach at our university. The last slide that I would like to show you is that I'm happy to see that our list is in accordance with the initiative called Vision and Change from the American Association for the Advancement of Science, in which they suggested that evolution, pathways and transformation and energy, informational flow, structure and function and systems should be present in every biology course, in every course to students. They need to understand those concepts. So I put colored arrows in each of one of those topics. So evolution, we have in our list, evolution of cell biology, transformation of energy, we have in the mitochondria and energy conversion topic. We have information flow, exchange and storage in cell signaling and communication and regulation of gene expression. We have structure and function, organelles in all those green arrows, and we have systems biology in genomics, transcriptomics, and proteomics that are included in these methods in cell biology. So this list we are now discussing at the Brazilian level with the Brazilian Society for Cell Biology, to try to stimulate professors to use this list to change their syllabi, including cell signaling, regulation of gene expression, and other topics that I listed here. And with that, I'd like to finish. And sorry if I took too long. Thank you very much for your attention. And I will be glad to take some questions if we have time. Questions could be either in English or Portuguese. Just one second. Okay, Miriam?
Okay.
Okay.
Perfectly right, but I'm not sure if you have questions from the chat.
Okay, we can, I don't know if it will be okay, but we can now have Manoel's talk and have some relation to my talk. At the end, we can discuss our talks together. What do you think?
No problem.
Yeah. Okay, Manoel?
Okay.
Okay.
Do you agree, Dr. Klymkovsky? Oh my God, I have a problem with your name. I have. Oh, it's a challenge for me. One day.
Well, I believe it. I mean, I think it's a very interesting list, right? And it captures most. I think you can teach. I mean, I think that's a very complete list. And the question is, just from my point of view, how you present it. So I like that list a lot. It seems very reasonable, but you're always talking about, well, I want to know this because I want to do that.
Yeah.
That's, I mean, so like, why do I care? Because, and so to me, it's sort of like, you could do almost anything you want as long as you have a good reason to do it, right?
Right.
But I think this is a very good. I mean, this is a very good core list. So it's a good thing to build on, right? Because you can easily, these things can explode outward, and the key is, because you have limited amount of time, you want to make sure that you're focused. That when you present it, the impact is maximal.
Right. We had a lot of problems with the intersection. I don't know how to call it. Superposed with biochemistry disciplines that we had in Brazil. Biochemistry separated, sometimes molecular biology separated, genetics, and the professors that coordinate these courses, they argue that sometimes they think that, for example, biomolecules, they should teach and not the cell biology teacher. The same goes for the regulation of gene expression. It's another battle. People from genetics or molecular biology classes, they think that they should teach that. But I think that there is a cell biology way of teaching, for example, regulation of gene expression. We teach in a different way as a geneticist will teach. We teach showing the different compartments, the different organelles, why a transcription factor, it's either like beta-catenin, it's either at the plasma membrane or at the cytoplasm or within the nuclei. And the molecular biology way of teaching this is not the same of the cell biology. At least it's the way I think.
I think that's exactly right. And so the question can be more of your theme, which is, are the sites along the DNA accessible? Are you talking about chromatin condensation? Are you talking about how the chromosome interacts with the mitotic spindle? How molecules are sequestered in and out of the nucleus without worrying particularly about specific transcription factors or specific genes
involved in the process.
Exactly.
Well, and even in the modern world where you're doing single cell analyses, you start to realize that in many cells only one of the two genes is being expressed. So you get what's called monoallelic expression, right? And so you start thinking about, you understand that at a cellular level, right? You understand it as these processes going on the cellular level, because it could be that this one gene is then condensed, the other one, is inaccessible, the other. So you could even, I mean, these disciplinary border wars are tedious, but you could focus on something more like, the general idea of what makes a gene expressible, what makes a protein, go back and forth. But, so you're not worried about particular genes and particular transcription factors. You're working about that general, how do I get access to the DNA?
Yes, exactly.
And that could be a way of making a boundary, if you want. I mean, I understand your frustration.
Yeah, it's a long time frustration, right?
I think sometimes it's the major challenge is to match the amount of knowledge and how this teacher knows about so many topics For example, when I was an undergraduate student, cell differentiation, because I'm graduating in pharmacy. Now, I'm not biologist. So, for example, the amount of hours in cell biology, it was very short. So, for example, to understand, to learn about cell differentiation, developmental phenomenon, it was very, very constricted. And I thought that I was able to understand this when I was in embryology classes. But now the emphasis was another.
Right. Yeah. But I think that cell signaling is really important. And the majority of the syllabi that I received didn't have cell signaling at all. So I think that this is something that we should worry about, right? They are not, the students are not receiving information or knowledge on those cellular processes. I think that that's what I get from that. It's more like a descriptive discipline and not something more dynamic.
Yes,
right?
Yes, that's right.
I hope we can change that.
The connections with other organelles,
yes.
It's another game.
Yeah. So, okay, maybe we should move on. Thank you.
Yes, of course. Let's talk with Dr. Costa. I will introduce Dr. Manoel Costa. He's a full professor at the Institute of Biomedical Science at Federal University of Rio de Janeiro. Biologist, during his doctorate, he worked with Dr. Carlos Chagas Filho at Biophysics Institute and with Howard Holtzel at the University of Pennsylvania. Afterwards, he worked at Oswaldo Cruz Institute and University of Pennsylvania during his postdoctorate. He is a specialist in optical microscope and image processing, having taken the optical microscope course at Woods Hole Marine Biological Laboratories in USA. He teaches cell biology for undergraduate courses in medicine and biomedicine since 1993 and belongs to the graduate program in morphological sciences. Dr. Costa's research focuses on cell differentiation, mostly in the myogenesis of zebrafish, a model in which he is a pioneer in Brazil. He has published the book Cellular Biology Manual and has maintained a website for scientific diffusion with histological images since 1997. He has published articles on science and education in the widely circulated newspapers such as Globo. Dr. Costa was my cell biology teacher 20 years ago, and I'm very grateful for this.
Okay. Thank you, Miria, for the introduction. Are you able to see my screen?
Not yet. Yes, now.
Okay. So I don't have much time left. I have to talk a little bit faster. If I became incomprehensible, please let me know. I add my bad English accent to try to talk everything so fast. So the first point is that I also don't have, sorry about that. Okay. So just to point out that my background, I do research on, as Miria said, in cell biology, but I have been teaching for a while. And I also did some work producing material, didactic material. But a few time ago, I began to I took part in a reform of medical course. And that was when I began to realize that there is really a lot of research going on in education. And I became fascinated by this research. And I tried to begin to do research by myself on education. And I tried to show a little bit what I have been doing since then. Basically, what I'll talk about is first, you heard a little bit about the core concepts or the overall concepts that should be present in any course in biology area as evolution, thermodynamics, information systems, and relationship between structure and function. And then Cláudia talked about work we did on this topic list on the intermediate, say, level, or this is not as concepts that are not as broad as evolution, thermodynamics, but also there's still a lot topic that has included several other smaller topics. I'll try to talk then a little bit about these individual concepts. How can we map these individual concepts? I think as Professor Klymkovsky began his talk, the question that drives us, I think the three of us begin by asking the question, what should we teach? It's not exclusive to us. Any professor should at some point ask himself what he's supposed to teach. And if you see a class having the expression that these students have, you really should think that there's something wrong going on. So I'll talk a little bit about this idea of trying to list which concepts should be taught and use of concept inventories, how we can try to map these individual concepts, the use of textbooks, and I will end in a completely different way, or actually in the same way, of trying to find individual concepts, how we can use these individual concepts. So I put this reference just to say that I don't advocate that we should be teaching a lot. Maybe we should teach less, and we should be teach more by teaching less. So the first thing I want to talk about is what has been really quite discussed in the literature is the idea of concept inventory. And I put here 3 definitions of concept inventory. I would let you read while I'm talking about them. But this is really a jargon for something that has been developed in the beginning in the physics field. They try to build a specific list of contents and to use this list of contents to assess how much students know about this particular content. So I will argue that this first use of this concept list, this concept inventory, is to use it in assessment. And this has been really discussed in the literature. I will also leave the PowerPoint presentation here if you want to use get the reference afterwards. And I point here that this has been extensively discussed. There are several models on how to do, how you should use the concept inventory. And first, how you should do the list of concepts, and then how you should ask this, if the students know this list of concepts. But actually, it's more than just to ask if the students do know or not know. Should also ask why they don't know, or why they get it wrong, and what are the misconceptions. This is a large field of research. Dr. Klymkovsky talked a little bit about this study of misconception in which the concept inventory have been also based. I did some sort of concept inventory in my course, in one of my courses in Rio de Janeiro Federal University, but not in this same way. I couldn't get the same level of depth. This is a course that is given by not just by me, but several professors to a given course, which is not my responsibility. So I couldn't change that much. I just asked the students, what do they know about a given concept? And I give the same test before and after, just before I begin the course and after I finished the course. The blue line represents, each graph represents one individual. The blue line represents the individual before beginning of the course, and the red line represents the individual after the course. The baseline would be they don't know anything about this, and the top line would be I understand completely this topic. So I just wanted to have the feeling of how the students felt about this kind of information and ask them about cell signaling, gene expression, or membrane, all topics, a list of 30 topics in cell biology. The basic idea here is that since we have courses that are have very different difficulty to selection process in the university, and some are much more difficult than other ones, if you want to compare how say a teacher performs or how students perform in a given class, it's not enough to give the same test, the same exam for all the courses. Since the students from the courses come from different background, it should be expected that the students that come from a more selective background know more than the students that come from a less selective background. So the question should be asking is, what is the gain of knowledge? What's the difference between the students, what the student knew at the beginning of the course and what the students know at the end of the course? So basically it will be this red line will be the difference or how effective was learning. And we can see, for instance, for this student, he didn't know too much in the beginning, but he ended up by learning a lot, or at least feeling that he knew a lot. Another use of the concept inventory is to use this process of building the concept inventory to understand, to ask what should be taught, and to learn about course design, and to adapt the course to this summary of what should be taught. And here I put some references. Just for instance, one, for example, this one talks about a framework or a hierarchical view of the course, as we talked about this topic list and the subtopic list, which will help us design a course and design a concept inventory for assessment. And here's another experience of developing a concept inventory as a tool for assessment. And it's interesting because they mentioned that they learned a lot. The students grew a lot by trying to list this, just to do the list of concepts that should be asked. And there are several biology concept inventory. There are few molecular and cell biology inventory. I didn't find any particularly just cell inventory of cell biology itself. Our work is also an attempt to do a collection of what should be known in biology. These maps of knowledge maps can also be not only listed, put in a list, but also as a map. And there's a very interesting literature about how to relate the concept to each other. There's even a very interesting software, the CMap tools, that allows us to do a map between them, to link concepts and to do a hyperlinked map of knowledge and how this concept relates to each other. Another thing that should be taken in consideration when trying to build this kind of list is that not all concepts are equal in abstraction. Some are just factual knowledge and some are more abstracted knowledge. This concept put up by Benjamin Bloom, that concepts have a hierarchy of abstraction, and you can go from a factual knowledge to comprehension, to application, analysis, synthesis, and evaluation. There are some modifications of this pyramid of knowledge. But the main point here is that the verb or the way you use each concept is different at each level. So the factual level, you have to remember, you have to memorize. And it's we don't use this expression. It has a bad connotation because all you have to do is memorize. So why do you need it? But actually, these are facts that the only relationship you can have to them is to either you memorize or not. Now, when you talk about, say, the theory of evolution, you're talking about something that is based on several facts and several interpretations. So this is a very abstract knowledge, and to understand this kind of abstract knowledge, you need to understand the other levels. So the point is the relationship between, to appropriate the concepts that are at this level are different from the effort you have to make to understand concepts at this other level. So taking all these into account, we try to set up a concept list of cell biology. You can think about this, Cláudia, Professor Mermelstein mentioned, the list of concepts at the topic level, and trying to make the list at individual concept level. So I try to list each individual concept, and I try to also classify it as either as function or structure or composition or evolution or just a historic knowledge. Of course, taking account that these are not exclusive. So something that is function, of course, is always an example of structure, but is also sometimes of composition and so on. And I try to list this concept as either a concept by itself or an example. And in this case, an example would be something that is just a way of applying this knowledge to explain in some case, but you could use other examples that would be as equivalent as this one. And also try to list some controversies or some concepts that are still not clear or people can argue about. For instance, there's a fact that actin filaments have 5 nanometers in diameter. Sometimes you can read in the book that actin filaments have between 4 to 7 nanometers in diameter, but actually there is a variation. Sometimes people mention that there's a variation in the size of these filaments. And the variation could be either caused by the nature of the actin filaments or the biological conditions which the experiments are made, and by the conditions which the experiments are made, or just could be artifacts. So I tried to list all these different concepts, and I came out with several lists. This had the help of undergraduate students, Maria Nabuco and Beatriz Ribeiro, and we came up with the list on the total. It's an ongoing process. We already have 340 concepts listed and classified in each category. I asked, for instance, I also asked how important are these concepts? If it's expected this should be known by any student in any course, these are fundamental concepts that need to be known by everyone, or is concepts that should be known by graduate students in the specialized area. How can we use such a list? And way of using such a list, for instance, we tried in graduate students, is to we ask the students graduate students have to teach classes, and we help them to teach classes. And the way we ask them is if you gonna give a class on a subject, you need to put up first a list of what you want to teach, and then you can do the slides and prepare the slides. And you can try to correlate your slide with the individual concept you want to map or you want to teach them. So I think it's more fairest way to try to identify what should be taught and what should not be taught. I noticed the time, I have to hurry up a little bit. I just mentioned that we talked about this idea of teaching in meetings in the Brazilian Society of Cell Biology. I included in this case the sixth concept, core concept, which is cell theory. And I used these 6 basic concepts and the list of concepts I made to develop an introductory cell biology class, which the semester, all the classes are being virtual or being given in the internet. But since I cannot change the curriculum, I offered this as introductory class, cell biology class, to all courses. It's available in the internet. If you want to use them, you're welcome to use them. And I move on a little bit fast. Just to mention that in some sense, the books are also collections of concepts, just that the concepts are not explicitly listed. And I think Professor Klymkovsky talked about his own cell biology, molecular cell biology book, or how can help the students to learn. And they also can talk about teaching without a textbook, or what's the importance of a textbook. And one of the ideas here is that I develop my little bit, not properly a textbook, but just a list of concepts that we think should be taught to the students. And I list the topics here, but I here explain a little bit further each topic, but I keep them as a topic. So it's very easy for the students to find what they need to know. and to try to be more effective because they know it's expected and that they understand. So in the 8 minutes I have left, I will talk a little bit about the last work I have been doing, is to try to go further down on the individual concept. Can we divide an individual concept? And the idea is to try to look for each word in a concept or in a sentence or in a group of words in a literary text. It's just the opposite of the plagiarism study, for instance, where you try to find this, what's the importance of a sequence of words. Here I'm just looking for the frequency of words. And I'll be treating the frequency of words as we would be treating the frequency of a gene in a proteomics or a transcriptomic studies. We did these studies where we studied the transcriptional changes during a treatment in muscle cells, and then we can see that some genes have a higher change in frequency, and we expect that this higher change in frequency has some meaning, for biological meaning for the cell. In the same way, I have my DNA tested, and I know that I have about 63% of origin in the Iberian Peninsula, and this is based on the frequency of genes that I have, and this frequency of positions, snipes that I have is typical of this region. So I use this idea, this concept, to try to measure the gap between cell biology and medicine. The reasons that the name of the chair is Frontiers in Biology and Medicine, and I teach cell biology for medical students, and we can see, and also during the reform of the curriculum of cell biology, of medicine, we have been also always asking these questions, that the problem that we have a basic cycle and we have a professional cycle, and these two parts don't talk to each other, and the students don't understand exactly what they should learn in the basic cycle if they only apply the knowledge in the professional cycle. So I asked the question, is this distance between basic cell biology and medicine, the gap between them is widening or is getting smaller along time? So I did this work with Cláudia Mermelstein and the undergraduate students, Sara Azevedo, Mariana Seixas. And what we did is we pick up 6 journals, 2 basic cell biology journals, 2 medical journals, 2 translational or experimental journals, and we used the Wordle and Voice Viewer software to try to analyze the frequencies of words. So here we have the frequency of words in the Journal of Cell Biology, and you can see that some words like cell and protein are much more common, much more frequent than others. And we can build a Wordle map, a word cloud map for all the journals, and it's easy to notice that the word cloud map for Journal of Cell Biology and Journal of Cell Science is very similar, while the map for medical journal is also similar. You have patient, you have letter, you have treatment, and the word map for experimental or translational journals is intermediate between these two. This word map only gives you a proportional visualization of how frequent the words are, but here are the list of words with their in the order of frequency, and you can see how similar is the distribution of most frequent word for cell biology between them, for medical journals between them, and for intermediate, for translational journals is somewhat intermediate between these two. We can plot these words in a Venn diagram, and we can see that there's a lot of words that are common. There's a common word core for medical journals, a large number of words that are highly frequent in both cell biology journals, and a very few words that are frequent, that appear as highly frequent in all four journals. This somewhat represents the situation, but the Venn diagram doesn't take into account the frequency, specific frequency of each word. For instance, cell is a very frequent word in the cell biology area, but not as frequent as in the medical area. So we developed what we call a similarity index that compare each word across several papers, and we summed up the number of repetitions, the frequency, and the proportion is of the total identity, the maximum identity possible. So the overall results are shown here. You have a 93 to 94% similarity index between cell biology journals. You have an 80 to 83% similarity index between medical journals. You have a 67 to 69% identity similarity index between translational or experimental journals, but you have a very small amount of similarity between cell biology and medical journals that are listed here and here also. We could even ask, is this gap becoming smaller with time now that there's a lot of translational science and translational research? What we did is to ask every 10 years. So we asked a long time, how does the frequency of word change? And you can see that cell is the overall most frequent word in all times, but some words have gone down, and structure was a very common word in 1965 and became less frequent with time, while words such as mechanism have become more frequent nowadays. And I highlighted here, I put in bold here, the words that represent processes such as function, mechanism, formation, interaction. These have become much more frequent in recent times in cell biology journals. The same goes for Journal of Cell Science. The same does not happen in medical journals. While some words are very common, it's not clearly that we couldn't find a clear pattern of if the words are going up or down, or which way are the words changing a long time in medical journal, in both New England Journal and Lancet. And in the Journal of Experimental Medicine, it's somewhat intermediate in the sense that some, you can see that, for instance, the increase in the frequency of mechanism, but we also have words like such as mouse that are frequent all the time. So we applied this to the similarity index calculation. What we came up was the similarity between, say, Journal of Cell Science and Journal of Cell Biology in 75 was 68% and is now of 95%. And the same goes for Journal of Cell Biology with Journal of Cell Science, that came up from 69 to 87%. This, not only the vocabulary was already similar, but is becoming even more similar with time. When you look at the medical journals, the similarity was quite high, but it's almost the same, as a little bit, went a little bit down. But if you go for the vocabulary similarity between cell biology and medical journal, which was quite low in 75, is now of only 40% and has been going down in all cases we look, which means that the vocabulary is actually growing further. It's becoming each time more different than in cell biology than in medicine. We are just submitting this paper. Let's see if it's accepted. How can you apply this for the study of cell biology and textbook cell biology? What we did is we analyzed the frequency of words in the major textbooks. These are the word clouds of the cell of molecular cell biology and molecular cell from Cooper. And we can see that more or less the list, again, cell is the more frequent word and protein is quite frequent, but there are some differences. And there is individual, particular vocabulary for each journal. You can see here that while there are lots of words that are common in all the three textbooks, some words are more frequent in Lattes, other words are in Alberts, and other words in Cooper. It may be possible that these unique words, or these words that have a higher frequency only in Alberts, say, represent an Albert style, an Albert approach, and difference, and we have to do further analysis to understand why these journals behave this way. The last thing we calculated is how similar they are between them, and we have a very high similarity index, which more or less means that they are equivalent, or we'll be talking more or less about the same thing. And also we tested if they're similar to journals cell biology and medical journals, and we can see a high value of identity between all the three books with the cell biology journals and a low identity indexes as would be expected between medical journals. We could ask, for instance, how do the textbook of the frequency of words in a textbook, such as Albert, relates with the change in time of frequency of word. I would expect that the textbook translates, I'd say, 10 to 20 year difference, which means that there's a new word, a new concept has been more frequent nowadays, and it takes a while for the textbook to incorporate this change in frequency. So I will stop now. We're already running over time, and thanks for the patience. And take questions if we have like 10 minutes or so for questions, if there is any.
Actually, I'm not seeing myself. Yes, yes. Congratulations for your talk. Anyone have any question? It's everybody shy here, especially today. But if you anyone prefer, you can write and we translate. Can write in Portuguese or even in English. It's not a problem. It's this. It's solving the problem.
Hi, I'm Arnon. Thank you, Manoel. Thank you, Cláudia and Mike, for the very nice talks. I'd like to ask Manoel, you just show us the difference. Well, I mean the similarity of the indexes about the books and the journals. Can you somehow measure the impact of this correlation? What is the meaning in practical means for this high similarity or this low similarity if you consider the journals, please?
I don't know. When I did the analysis in the journals, medical and cell biology journals, I could compare because I have an internal control. So I guess the best way to assess this would be comparing different books that are not from cell biology, textbooks that are not from cell biology, and see if they're similar or not to the journals. I'm not sure even if it's a high similarity index should be expected, in a sense that is it good that it's very similar to the journals? It may mean that if it's really similar to the journal, it's not didactic, it's not a good way of students to learn. But if it's too different, it does not reflect what's the really hot topics in the area. So I would expect that there's some correlation, but not a direct correlation between a high identity index between those two. I would like to analyze a long time. This would be interesting to see if they're getting more similar or less similar. I think that somehow the books are getting into a dead end. I mean, the Albert, the last edition of Albert, it's not, you can't access it in printed, you have to access it in a CD because it's too large to be printed. So this is really too much knowledge, and maybe it's just too much to know. And one thing is if it's accurate enough, it's similar enough to the journals. But this is measurable. But how efficient it is in teaching, it's not. I can't measure it.
Thank you.
Okay.
Any other questions? Can I finish the section?
Mm-hmm.
Yeah. Professor Klymkovsky, I really appreciate again. I really appreciate again your presence here with us It was a pleasure to talk to you and Professor Mermelstein and Professor Manoel Costa and everybody that is here during this pandemic year. It's not easy to be to stay for a long time facing a computer and discuss science. Yes, because it's exhaustive, and discuss science, discuss other ways to teach and learning about science, cell biology, biochemistry, everything. So I have a note, something that Manoel told me about the next meeting is the 22nd in this month, and the time it changed because our I forgot the name of Rita. It's Rita, the name of her. She's from Portugal, and the hour is too late for her. So we changed the hour for 3 p.m. just in this day, okay? We're talking about advances in medicines and biology, okay? Do you have any other things that you'd like to talk, Manoel and Cláudia?
Just to thank everybody.
So yeah. I thank you again, everybody.
Okay.
Thank you for the invite. It was fun.
Thank you.
Bye.
Bye-bye.
Yeah.